Hi everyone. Today we're going to examine a recent study that uncovers a rather intricate metabolic dialogue between colorectal cancer cells and the seemingly normal tissue that surrounds them.
So, we'll proceed by first outlining the clinical observation that started this whole line of inquiry. Then, we'll deconstruct the molecular mechanism piece by piece, which reveals a novel feedback loop. And finally, we'll review the in vivo validation that ties it all together.
All right, so the investigation actually begins not within the tumor itself, but in what's called the normal adjacent tissue. We're talking specifically about the colonic epithelial cells, or CECs. Now, these cells have often been considered passive bystanders in the progression of cancer, but this study really challenges that assumption by investigating their active role. The central question becomes, what is the nature of the crosstalk between these non-malignant cells and the tumor right next door?
Well, an analysis of clinical cohorts revealed something quite specific. A protein called growth differentiation factor 15, or GDF15, is significantly upregulated in these peritumoral CECs. And this isn't a minor correlation. GDF15 expression increases with advancing tumor stage, and perhaps more importantly, it strongly correlates with reduced overall patient survival. The key finding here is that this GDF15 is predominantly secreted by the peritumoral CECs, not the tumor cells themselves. These GDF15-producing cells also display markers of senescence, a state of irreversible growth arrest, which suggests they're in a distinct, altered state.
So, the next logical question is, what is the functional consequence of this elevated GDF15 secretion from the surrounding tissue?
And what the in vitro experiment showed was that this secreted GDF15 acts directly on the cancer cells. When colorectal cancer cells were exposed to media conditioned by these GDF15 secreting epithelial cells, the proliferation was significantly enhanced. Functionally, GDF15 appears to induce a significant metabolic shift in the cancer cells, moving them towards a highly glycolytic state. This is evidenced by increased glucose consumption and a marked rise in lactate production. Now, this effect is abrogated when a GDF15 neutralizing antibody is introduced, which confirms that GDF15 is indeed the driver of this metabolic shift.
The investigation then honed in on the mechanism and identified a specific glycolytic enzyme, enolase 1, or ENO1, as the key mediator. And critically, when they went back to patient samples, they found a strong positive correlation between the GDF15 levels in the normal adjacent tissue and the ENO1 levels within the paired tumor tissue.
So let's trace the precise molecular pathway inside the cancer cell. How does that external GDF15 signal translate into increased ENO1 expression?
Okay, the pathway initiates when the secreted GDF15 binds to its cognate receptor, GFRAL. This receptor is expressed on the surface of the colorectal cancer cells, and this specific binding is the crucial first step.
This receptor-ligand binding event then triggers an intracellular signaling cascade, which begins with the phosphorylation and subsequent activation of the JNK1 kinase. Once active, JNK1 in turn phosphorylates STAT3. Now, STAT3 is a well-known transcription factor, and its phosphorylation is a necessary step for its activation and its eventual function inside the nucleus. This phosphorylation event leads to STAT3's translocation into the nucleus. Using chromatin immunoprecipitation assays, the researchers confirmed that STAT3 binds directly to a specific site on the ENO1 promoter. This binding drives the transcription of ENO1, providing the molecular basis for that increased glycolysis we saw earlier. But this one-way signaling from the normal cells to the tumor cells doesn't quite explain the initial observation, which was the sustained high level of GDF15 in that peritumoral tissue. So, the investigation pivoted to address this. Is this just a one-way street, or is it possible that the cancer cells send a signal back to the epithelial cells, creating some kind of self-reinforcing loop?
And the mechanism they uncovered involves a process called histone lactation. This is a relatively recently discovered form of epigenetic modification where lactate, the end product of glycolysis, is actually used as a substrate to modify histone proteins, which can directly influence gene expression.
This is how the complete feedback loop is formed. The highly glycolytic cancer cells export large amounts of lactate into the microenvironment. This lactate is then taken up by the peritumoral CECs through transporters like MCT1. Inside these CECs, the lactate serves as a substrate for H4 lysine 8 lactation, specifically at the GDF15 gene promoter. This epigenetic mark then recruits the writer protein EP300, which activates GDF15 transcription, leading to more GDF15 secretion. And of course, that secreted GDF15 then simulates more glycolysis in the cancer cells, which produces more lactate, perpetuating the entire cycle. So to confirm the physiological relevance of this entire axis in a living system, the researchers turned to orthotopic mouse models, where human cancer cells are implanted into the corresponding organ, in this case, the colon.
By using conditional knockout models, they were able to selectively delete key genes like GDF15, but only within the intestinal epithelium. This level of precision is what allows them to isolate the specific contribution of this pathway in these cells to overall tumor progression.
And the results were quite clear. The genetic disruption of any key component of this pathway, whether it was GDF15 itself, its receptor, GFRAL, or the downstream kinase JNK1 in the intestinal epithelium, all of these interventions significantly attenuated tumor growth, tumor load, and lactate accumulation. These findings provide strong in vivo evidence that this axis is essential for colorectal tumor growth.
This research really elucidates a previously unknown mechanism of collaboration between malignant cells and their so-called normal neighbors. The authors conclude that this crosstalk provides a potential new avenue for targeted intervention. This mechanism challenges the conventional view of those peritumoral cells as just passive bystanders. It reveals them as active, senescent participants that are effectively co-opted by the tumor to help orchestrate a procarcinogenic niche. So, to put this into perspective, the conventional view often considers peritumoral cells as passive tissue and tumor growth as something driven primarily by intrinsic factors. This work revises that model. It demonstrates that these peritumoral CECs are senescent and actively collaborate in a reciprocal feedback loop. It also directly links a metabolic byproduct, lactate, to a specific epigenetic change, histone lactylation, that fuels the cancer's progression. The discovery of this druggable GDF15 lactate axis deepens our understanding of the tumor microenvironment, and it also presents a potential new strategic target for therapeutic intervention in colorectal cancer. The question that remains, of course, is how effectively this axis can be targeted in a clinical setting to disrupt this metabolic and epigenetic symbiosis.